This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 11, recorded on July 8th, 2011. Hi, everybody. I'm Vincent Racaniello, and this is TWiM, the podcast that explores unseen life on Earth. Joining me today from the Medical University of South Carolina, Michael Schmidt.
Hello, Vincent.
Good to have you back again. How is the weather down there in North Carolina?
It's as expected. It's summertime, and everybody's moving slowly because the humidity is high, as is the temperature. So
that's too bad. Well, here we have clouds, so not a nice day. Also joining us today, moving westward from the University of Wisconsin at Madison, Margaret McFall-Ngai. Hi, Margaret.
Hi, Vincent. How are you?
I'm fine. Welcome back.
Thank you.
2 weeks in a row. That's great. you must like it.
I do.
Good.
Do. And
finally, we're going to go to the West Coast and find Elio Schaechter. How you doing?
I'm doing great. I'm becoming a Californian by the minute.
Yeah, you've been there for a while, though, haven't you?
Yeah, I have about 17 years now.
Oh, you're already a Californian, no?
I am. I've been here as long as most people.
Yeah, well, California has a lot of East Coasters, right?
It sure does.
Yeah, that seems to be a very attractive place. I almost went there as my first job, but I decided I couldn't move to California.
Okay.
After having lived on the East Coast all my life. I don't know, the palm trees were too much for me. Today we have two cool papers to talk about. We had hoped to have Jo Handelsman here with us, but she is nowhere to be found, so we will proceed without her. The first paper is a paper that came out in Emerging Infectious Diseases just recently, and it's called Extended Spectrum Beta-Lactamase Genes of E. coli in Chicken Meat and Humans, the Netherlands. And I asked Elio if he could give us a little summary of this paper.
Well, the general subject is that we may be acquiring nasty bugs from animals, from farm animals, and that this may well have to do with the use of antibiotics to promote growth among the animals. In other words, in this country, it's a widespread practice to use antibiotics in the feed. And the idea is that it may or does increase the growth of the animals. This has gone on for a long time. This is an old subject.
They give low levels, right? So
they give low levels,
and that's the worst for selecting.
Right, because
resistance.
You don't kill the bug, you just make it resistant. So this paper is one of many that establish an epidemiological link between certain strains, in this case of E. coli, in chickens and in people. Now, the reason this paper is important is because it's being done in the Netherlands. And the Netherlands are an interesting place with regard to this issue. The reason being that they use a lot of antibiotics among animals for feed, unlike other European countries that have scaled back or cut it out altogether. We'll talk about that a little bit later. But in addition, the incidence of strains like this until recently among the human population was very low. And the reason for that is because even though they use antibiotics for the animals, their use in humans is very, very restricted. You don't just prescribe antibiotics and get them. You don't go into the doctor's office and say, hey, doc, give me a shot. I have a flu. I have the flu, give me penicillin. That's not done there. And so the measures they have taken which limit the amount of antibiotic use. So this means that there is not a great deal, or there hasn't been until recently, a great deal of antibiotic resistance in strains of humans. This has changed. And this is why the study is important, because it's done against a background different than that, for instance, in the United States, where such a study would be very difficult to carry out at a meaningful level, because it's just really a correlation. It's an epidemiological correlation. But here you're starting at a baseline, which is far, far lower. So here's the story. They went to 4 hospitals in Holland, the Netherlands, and over 2.5 months in 2009, they isolated E. coli from patients by using rectal swabs. And at the same time, they went to the grocery stores nearby and they picked on ready-to-eat meat, packaged meat.
This is raw meat, right?
This is raw meat, right. Unspiced and untreated in any way, just meat. And what they found is that in 45% of the patients, they found a strain of E. coli which harbors a gene for beta-lactam resistance. That is, this beta-lactams is, as you know, are penicillins and cephalosporins. And so this particular enzyme that's carried by this, encoded by this gene, breaks up the antibiotic, and the antibiotic doesn't kill the bug, and the bugs become resistant. The particular gene is called ESBL. I know you had enough of acronyms, but we'll have to use it. It stands for extended spectrum beta-lactamase. Okay? And there are several varieties of that. So what they found is that they didn't just take rectal swabs, they also looked at blood cultures. Anyhow, in 80% of the chicken samples, they found the ESBL carrying genes. Among humans, they found quite a few. I have to look and see what the number is, but it was present in 57% of rectal specimens and 57% in the blood cultures. And we can go into a little bit, but the point here is that they did the correct analysis, at least I think they did. They looked at the sequence of this gene, and they also did something else, which is called multilocus sequence analysis, or multilocus sequence typing.
Typing.
Yeah. If I may, I'll explain what that is. If you want to determine the homology degree, or the degree of relatedness between strains, you can pick on a gene. You could pick on the small ribosomal RNA, or you could pick on something else. You could pick on almost anything. But the better way of doing it is to say, let's do it for 6 genes. And there are 6 genes which are sort of standard. They're used a lot. They are housekeeping genes, a variety of things that are just sort of present in any bug, different from one bug to another. So by doing that, you have a greater confidence in establishing homologies. So that's the story for now.
Can I ask you, the patients where they got samples, these are randomly selected people, right? Not people with particular infections, right?
I'm not sure I know, but I think that's right.
Yeah.
I don't know that they said. Random, yeah, well,
they get 900 and some rectal swabs, and then they say
that's right.
59 of them were most likely infected with ESBL producing E. coli. So
correct.
So a subset of those were. But even in the individuals who were not infected, they had.
That's right.
They had some of these genes, these resistance genes.
Yeah. So all of a sudden, the Netherlands are no longer the paradise.
Safe haven.
The safe haven.
So this figure 1 is the telling figure where they have 3 pie charts.
That's right.
And they have these beta-lactamase genes just come in different flavors, right? They're all different kinds of specificities, and they have them all here. And the biggest one in chickens and then the rectal swabs is CTX M1, I believe, if I can see the color. Is that correct?
Yeah, that's correct.
And so that means it's the same plasmid-borne gene, right?
That's right. And if you add to that the multilocus typing business, it's probably the same bug, as close as you can get.
And if you look at figure 2, which has got the network, because what multilocus sequencing does for you is it shows how the microbe has moved in a general geographic area. That's the beauty of being able to sequence housekeeping genes. And what it effectively does is it asks for genetic drift, if you will. And you can then plot by differences in these very small, it's a small PCR product that they sequence, and they ask how many bases are different between the sequence types, and that then gives them a number. You can see it almost looks like a map of a town.
So this map tells you the strains.
It really drives home
They
the
go
point
together.
that was made about how paradoxically you have the use of antimicrobial drugs in the farm production animals in the Netherlands, and then you have the lowest use of antimicrobials in all of Europe, also in the Netherlands. But the map, the multilocus sequence map, is really arguing that, well, the drug resistance traits are moving from the farm to the table to the individual. And so they'll have the same issue with antibiotics no longer being as effective when they're needed to treat infections, simply because of the foods that they're eating and how the foods have been grown with these antimicrobials to effectively shorten the time from farm to table.
Let me clarify this because I may have misspoken. So 80%, 79.8% of the chicken meat had these ESBL genes. And then they say 5% of the hospitalized patients were also carriers of ESBL genes. So I take that to mean, Michael, and you correct me if I'm wrong, the carriers, they didn't have disease, but they were positive for these ESBL genes?
Right. It's become a component of their normal flora. Okay. And that's the issue with the whole infectious paradigm. Oftentimes, the infections that get us in the end are our own flora. You know, our resistance breaks down. We have penetrance due to our gut. We have a breakdown where the microbe gets introduced into a sterile body site, and the net consequence is then an active infection with our normal flora, as opposed to a healthcare worker transmitting that microbe from another patient or from an environmental focus to the patient.
One wrinkle which I found interesting is that the strain in question is so common in chickens and not at all common in beef and pork. So this is an issue that the Salmonella people love, namely to talk about what's involved in host range. That is, in Salmonella, there are strains which cause typhoid in people and don't do much in animals, or they cause a disease like typhoid in mice and not so much in animals. So the host range idea is coming to the fore here, because we must be a lot like chickens when it comes to this particular E. coli.
So and cattle and other types of animals are also treated with antibiotics, right?
Right.
All right. So
there are probably other problems. I mean, this is an E. coli study. You can do the same thing with Campylobacter, a pretty nasty bug that causes diarrhea, and you can do it with Salmonella. So this is endless. I mean, this is just one study.
Sure.
And who knows what will come out if you looked at the bugs.
So what's the problem about having these ESBL genes in 5% of the population? So these people are healthy, right? When does it become a problem?
It's really an exercise in population dynamics. It's how the broad spectrum antimicrobial genes move amongst the microbial world, because as we well know, microbes like to exchange information, and in the proximity of the human gut, if you're carrying, and that's where they got 5% of the carriers had the ESBL in their stool, and the net consequences, those ESBL genes could then be exchanged into, say, for example, one of the microbes that we talked about a few weeks ago, the famous E. coli that's now running through Germany. And it's these movements of these genes, and you go back to figure 2 in this paper, illustrating the genetic networking of how these genes move from the chicken meat into other meat types as well as into the human population, that really illustrates the complex dynamic of what's going on with these multi-drug resistant genetic material that's causing havoc in the medical community.
Let me clarify something, Vincent.
Sure.
Although only 5% of the normal people carry this bug, when it came to looking at blood cultures, it was 74% of the patients. It was a smaller number, of course. It was 31 blood cultures, of which 23 were positive. Now, they don't say why these people why they drew blood of them, but I imagine that they were sick. You don't just draw blood of a healthy person. So whatever they were sick with, they might have been sick with this E. coli. That's not stated.
Right.
And I'm, something to wonder about. But, it's worrisome regardless for the reasons that Michael said.
Right. So these are reservoirs now of resistance genes which can be given to other species of bacteria.
Now, the issue about the use of antibiotics in food animals was very nicely written up in the current issue, the June issue of Microbe, that's the ASM house organ, by some people here in the United States and in Belgium and in Sweden. And if you want to look at it, it tells you exactly what several countries in Europe are doing about it: Sweden, Denmark, Netherlands, and United Kingdom. So what happened was that in 1986, Sweden and then Denmark cut down on the use of antibiotics as growth promoters. And now you ask, it's a legitimate question to ask, is it really true that antibiotics promote growth in farm animals? Because it's a major economic issue. And the answer is yes, it does somewhat. However, if you substitute the antibiotics for more advanced farming practices, where the animals are treated, I don't quite know how, but I'm reading this someplace, that if you treat them better, the difference between the treated and antibiotic treated animals and the non-antibiotic treated animals is very small. It does cost more. It has a cost to the meat.
Right.
So, this is not something that you can be totally indifferent to. However, it works. And in Sweden, Denmark, apparently in Germany, these measures have actually been, in the United Kingdom, these measures have reduced the amount of antibiotic-resistant bugs in the animals. Well,
that's how you do the study, right? That's exactly it.
Right. And in one curious situation, in the ruminant animals that we use for food, like cows and sheep, we know that if you treat silage, which is a fermented product, with the appropriate probiotic to effectively prepare the food for digestion by the animal, you can actually have twice as much weight gain if the food is properly conditioned with the probiotic as you can with the same food that was just treated with a natural ensiling process. So
Michael, are you absolutely sure of this?
I am. I actually did a research project with a group out of Iowa called Pioneer Hybrid, and they have empirically developed probiotic inocula for silage, whole plant corn silage, and alfalfa and other products, and their silage inocula that they place onto these ensiling materials actually facilitate weight gain in the animals using the same project. And they did controlled studies in which they ensiled material with their inoculum versus a control inoculum that was just left to naturally ensile itself. And they were able to show differences in weight gain.
Let me ask you about the use of the word probiotic. Probiotic to me means a bug that colonizes a host. Was this the case here, or was it simply the case that if you add bugs to the silage, they will break down the silage for the benefit of the cow?
It's the benefit of the cow. I misspoke. I used the word probiotic in its generic sense. I was considering probiotics colonizing the corn or the alfalfa to effectively prepare the food material for the animal. It's not, and obviously it's a live product that the animal eats, but I don't know if they have ever demonstrated that the microbes actually move through the gut of the animal and establish themselves in that way.
So, Margaret, having seen this, and this is probably something you don't pay much attention to.
I do. You know, so as I was listening to you guys talking, I was thinking about the fact that it just underscores our insensitivity as humans. I feel like I think I may have mentioned this before, that we've forgotten two things. One, that we're animals, and secondly, we're part of the biotic world. And so as such, there's a continuum between ourselves and the environment that we live in. And I think that, of course, we're going to be talking about our learning about the normal microbiota of humans. Well, certainly these issues of the normal microbiota and their balance apply to all other vertebrate animals for sure. We know that this goes all the way down to fish. And so you can see how growing animals in stressful conditions is going to upset the ecology of their microbiota. When Elio was everybody was talking about growing them under less stressful conditions and having a better outcome, that makes all the sense in the world to me. And so I think that that's my take on the whole thing, is that we need to start becoming sensitive to the fact that we're trifling when we give antibiotics anywhere at any time without understanding the very complex nature of our interactions with the microbial world.
Yeah, unfortunately, when it's a matter of profit, those considerations go by the wayside. Oh,
absolutely.
I wondered what the administration of antibiotics to all these food animals does to their microbiota.
Oh, that's what I mean. Yeah.
And
sure.
It must be disturbing them in some way. And maybe that's why they grow faster, is that you alter the microbiota in some way.
Well, that's one idea. In ruminants, of course, you worry about getting rid of the bugs in the rumen.
Yeah.
That's rather bad on a cow or a sheep. They would starve without the bacteria there.
Well, and
it's complicated. There are two ends to a cow. The rumen and the large intestine.
Well, one of the interesting things is that in germ-free mice, for instance, and other germ-free mammals that they've studied, it takes a third more food to keep a germ-free animal alive than what they call a conventionalized animal. In other words, an animal that carries its microbiota. So I find that interesting in relation to the fact that you're getting a larger yield when you give them antibiotics.
Yeah.
Does this have to do with the stimulation of the intestinal epithelium by the bacteria?
It's actually, the microbiota change. The presence of the microbiota changes so many features of of an animal, all the way down from its behavior, all the way down to the microanatomy and macroanatomy of its gut. And so, of course, oftentimes these germ-free animals are germ-free from birth, and so they are not allowed to mature and whatnot. And also when you treat an animal with antibiotics, there's a lot of evidence now that reservoirs of their normal microbiota remain in the mucus layers and so on and so forth. So what you're doing basically is you're knocking back the microbiota. You're not eliminating it with antibiotics.
Right.
And that's one thing that we haven't talked about from the perspective is it's not only the diversity of the microbiome that's important, but it's the relative number of individual species, because it's a genetic dose effect, because each and every one of these microbes is making product that are talking to our systems. And we know from our various earliest times in looking at good old E. coli and the lac operon, gene dosage is real. And we know from those of us who trained as molecular biologists know that we just love to death multi-copy plasmids because we can make boatloads of product. And as you begin to think about this complex and dynamic equilibrium in something as complex as a gut and the signaling that must be going on in order to put weight onto an animal, Gordon's group out of WashU has done some really elegant studies with mice showing that the microbiome actually determines whether or not you're going to be fat or not.
Yep. Oh, yeah. Yep. Yeah, absolutely. And we'll talk about that today with regard to Helicobacter.
Oh, yeah.
I mean, it's really, we are an ecosystem. And, with things like inflammatory bowel disease, they've shown that the diversity of the microbiota is much lower in a patient that has inflammatory bowel disease. So that high diversity seems to be an important determinant of health. Absolutely. You can see one day restoring that diversity as a way of treating infections rather than giving antibiotics, right? Absolutely, yeah. One of the other consequences of feeding these animals antibiotics, of course, is they end up in the environment too. Oh yeah. They go in the aquifers and so forth, and they have more effects than just on the animals and on us. So maybe we should point out that the way the bacteria get from the chicken to humans, I presume, is when you buy the chicken meat, it's raw, and you touch it, and you contaminate yourself right before you cook it.
That's how I would envision it, because I don't know too many people that eat sushi-flavored chicken.
No, and so it's inevitable because you have bare hands, right?
Yes.
And you're going to contaminate yourself.
The cutting board in your kitchen is probably the most dangerous piece of equipment in your house.
Yeah.
It's very buggy.
Yeah, it's more dangerous than the knife.
Right. So you get these bacteria with antibiotic resistance genes. They colonize you. You're fine until you need to have antibiotics for some other infection, and then you're resistant. That's the scenario, right?
Well, yeah, but who knows?
Or you spread it to other people.
The fact is, when you start introducing very effective plasmids that transfer the resistance genes, they can also transfer other things. And after all, this is we have seen the likely emergence of very dangerous bugs through a mechanism of horizontal gene transfer within historical times, maybe. So you can also acquire not just resistant genes, but you can acquire genes for toxins.
Sure, absolutely. So a number of years ago, the Pew Foundation commissioned a study on industrial farm animal production. This was, I think, in 2008. And I'll put a link to it in the show notes. It's called Putting Meat on the Table, Industrial Farm Animal Production in America. And it's a beautiful summary of this whole problem and how farm animal production has evolved in this country from mother and father farms to the present hyper-industrial state. And they actually they made a series of recommendations, and one of them was to phase out the use of antibiotics in animal feed. And the other was to give the animals better living condition, because as Margaret said, they're very stressed. They're in crowded conditions, and everything is mechanized. The feeding is mechanized, and the waste elimination is mechanized. In fact, you can have these thousands of head swine farms with only a few people taking care of them. And they argued that just by giving them less stressful environment would be better. But of course, I don't believe that any of those recommendations were ever followed through, unfortunately. Well, and for short-term profit, yeah. Yeah, that's the problem. So
they were in Sweden, but not here.
Yes. So here it's going to be harder. I don't know, people still don't believe in global warming. And here is something for which we have the data, so at least they're correlative, but you can imagine many more studies that could be done and which would provide the right data, yet we still wouldn't change our ways. So I found that very frustrating, reading through this and realizing that it's not going to change anytime soon.
Well, it's going to change as we lose the antibiotics that we use to treat infections as we become ill. And when we go back to the beginning of the 19th century data, when we recognize that a strep throat infection had a mortality rate of about 80%. And I think that's what's going to drive the situation when, you begin to lose your children, or you lose your parents or yourself to an infection simply because we no longer have antibiotics when we need them, because we've been using them to put food on the table more quickly. And that's going to be the decision that we as a learned society are going to have to help everyone think about.
Sounds like it's going to take some time, though.
Yeah.
And a real dearth of effective antimicrobials before it filters down to the food producing level, which is unfortunate. But Elio, any other things we should know about this study before we leave it?
Oh, I think you just should stay tuned, because there'll be a raft of studies of the sort coming up in different countries.
But as you said, you can't do this in the U.S., right?
Well, because you have a high baseline of resistance. So, if a lot of people carry resistant bugs which are resistant, you can't really attribute a bump in that number to anything. Very difficult to do. So it's in some countries it'd be easier to study than in others. But look, the thing is, it doesn't matter, because these principles are universal. And so what happens in Holland doesn't just stay in Holland, as they say in Las Vegas. It's going to be all over the place.
Yeah, but I can imagine the food companies arguing that, okay, this is what happens in Holland, but show it happening here in the U.S., right?
There'll be. I would be very surprised if something very telling didn't happen along a slightly different scenario, perhaps, than the E. coli one. But there are plenty of bugs that are emerging which are not present in the population in large numbers, and which are, if they pop up, if there's a bump in them, then the information will be available.
Well, I said to my colleague today, well, what it would take would be Frank Perdue dying of a untreatable infection, and he pointed out that Frank Perdue had already died. So
maybe he died of an untreatable infection.
It could be. Well, then his offspring will take over the business.
Right.
Okay, let's move on to our second paper. This was published in the Journal of Clinical Investigation. It's called Helicobacter pylori infection prevents allergic asthma in mouse models through the induction of regulatory T cells. And I thought this was a great segue to our discussion last week, Margaret, of the squid Vibrio symbiosis in a way.
Yeah, absolutely. So this paper was, I thought also, this is very interesting, right up my alley. And this is like you say, what I think about. The squid is a really easy model to study. But this Helicobacter pylori is a bacterium that lives with humans. And the article here had three components to it. There are three elements. The element of asthma, and it's called atopic or asthma, and atopic asthma is asthma that's the result of exposure to an allergen like dust mites or something like that. The bacterium, Helicobacter pylori, and the relationship between the two of those things, between the asthma and the bacterium, or the carriage or having with you as part of your microbiota, this particular microbe, Helicobacter pylori. So I thought that they did a really nice job, a succinct job with their introduction. They talked about the fact that asthma is a chronic inflammatory disease of the airways. You get constriction and wheezing and coughing and whatnot. And this particular type of asthma that they're talking about, this atopic asthma, which is due to exposure to allergens, is the majority of the asthma sufferers have this type. And so, 75% of them or something. And the incidence of this is staggering. And Americans, 1 in 10 children and 1 in 12 of the whole population. And this incidence is rising. And I guess over the last 10 years, there was a study, I think between last 8 years, 2001 and 2009, that showed that the incidence has gone up an entire percent. And so we're really increasing in our asthma incidence. And they talked about the reasons that biomedical researchers have, thought that why is this happening? And there are two major ones that have been given, and that is that there's increased pollution and increased exposure to cigarette smoke and the like. But in fact, I mean, think about it, fewer people are smoking, as far as I can see. And I don't know, Elio, you live in San Diego. I grew up in Los Angeles, and I can tell you that the air pollution in Los Angeles has gone down dramatically.
Yeah, certainly.
So that's likely not to be the case. The second thing is something called the hygiene hypothesis. And the hygiene hypothesis says that the lack of exposure to environmental microbes when you're a kid or, when you're growing up predisposes you to getting things like asthma and allergies. In other words, your immune system needs to be educated. And the way you do that, it's hypothesized that kids did that more, in the past because they were playing outside more. And there's some support for that idea. But two very famous microbiologists that they mention here, Martin Blaser at New York University and Stanley Falco at Stanford, have come up with a third hypothesis that they talk about in a very well done review in Nature Reviews Microbiology last year about the loss humans, because of in the developed nations, we are tending to lose components of the microbiota that we evolved with. And so Blaser and Falco feel that the loss of these critical components are compromising our health and likely making us susceptible to things like asthma and allergy. So one of these organisms is the topic of this paper, and that's the Helicobacter. Who's yawning there? Who's the Helicobacter pylori? So Helicobacter pylori. The audience may have heard about this as the causative agent of ulcers, gastric ulcers, stomach ulcers. And also it's been implicated in progression of gastric ulcers to gastric cancer. And it's interesting that virulence, or that those diseases caused by Helicobacter are in part mediated by carriage of certain virulence genes, one of which is covered in this paper. It's something called CAG-A, and CAG-A is a toxin. And so in the diseases, it's important that CAG-A be present as a mediator of the production of ulcers. Now, it turns out that, interestingly, we for a while we thought, oh my gosh, carriage of Helicobacter pylori is really bad. You're going to get an ulcer. Well, Martin Blaser has spent a lot of time working on this, and it turns out that individuals in developed nations, like the United States, have been losing their Helicobacter over the past decades, and that is through diminished transmission from mother to child, as well as the use and overuse of antibiotics. We've seen that in the beginning of the 20th century, the vast majority, like 90% of Americans, had Helicobacter. Asymptomatically, you just carried Helicobacter. And now, most people don't, only about 10%. And the loss has been correlated with the onset of many, many diseases. And so we think that Helicobacter, the loss of Helicobacter, may be causing things like acid reflux disease. So it's been correlated with this loss of this particular stomach microbe. Seems to be correlated with things like the onset of more acid reflux disease. And it seems like Helicobacter pylori maintains a healthy pH in your stomach. And also, as we were talking about a little bit earlier, Jeff Gordon had shown that the microbiota of the gut can be important in obesity. Well, Helicobacter pylori in your stomach balances the hormones in your stomach, and they're the hormones that are involved in signaling hunger and fullness. So if you don't have Helicobacter, there's a correlation between obesity and the lack of Helicobacter.
So is it mainly in the stomach or the entire gastrointestinal tract?
It's just mainly in the stomach. Yeah, It's a stomach bug.
And it loves staying there.
Yeah.
And it does all sorts of remarkable things with how it modulates its outer membrane and its LPS. It has a very elegant set of genetic controls, and it undergoes slip-stranded mutagenesis in order to maintain its genetic diversity while it's in your gut, fighting off our immune system. So Helicobacter has evolved to stay put in our stomach, yet for some reason we're able to, as a population, now dispense with it, which is really pretty remarkable.
Although we have all these correlations with the onset of diseases because it's not there anymore.
Yeah.
So I don't know if this strays too much, but under what conditions does the presence of H. pylori cause a gastric ulcer?
So, I think that, Marty Blaser says that generally it happens post-reproductive. In other words, after your years when you're having children. And so what that means is that as a genetic character, the ability to have Helicobacter, or the way humans carry Helicobacter, because you get the diseases post-reproductively, it can't be selected against in its carriage. And so usually you don't get ulcers until you're later in life, or gastric cancer until much later in life. And it's the result of imbalances and stresses, and things just get out of balance. And as I think Michael said, one of the things that happens in many diseases is that part of our normal microbiota goes to the dark side. And in that, this is the case with Helicobacter, and there's a lot of evidence. I mean, one of the most interesting studies that Marty Blaser did was to show that you can trace the migration of human populations by studying the strains of Helicobacter that are carried.
Right.
And it's just so there's a lot of evidence that we evolved with this microbe, and it's really important. And in the case of this paper, in addition now to correlating with acid reflux disease and obesity, there have been a series of papers correlating asthma and allergy with the loss of Helicobacter pylori. Now, obviously, you guys have noticed that in my descriptions of the possible effects of Helicobacter and pylori in humans, I've used the phrase correlates with.
Right.
And so there's the connection with this paper, because so much of the work done so far has said, humans are losing it, and at the same time they're gaining this thing. And so the cause and effect, whether or not Helicobacter carriage is important as a cause and not just something that's correlated with something else that's happening, is the major question. And so what this paper did was to provide a set of experiments designed to determine whether being colonized by Helicobacter would render an experimental model animal resistant to developing asthma. So, of course, you can't experiment with humans. So there's an experimental model of animals, and that's a particular strain of mice. So basically, they asked two questions. They asked, does the carriage of Helicobacter in an animal protect the animal from developing asthma? And the second question they asked is, how in the world, if it does, how in the world does it do it? And so for the first question, does the carriage of this microorganism by an animal protect it from developing asthma? What they did was they took lab mice of two different ages. They took newborn mice, and then they took adult mice. And they looked at each of these groups with colonization of Helicobacter pylori and without colonization of Helicobacter pylori in the stomach. And they found out in these cases what it did to the animal's ability to resist an allergen. They did a series of them, but one example is they used dust mites. And what they found was that mice infected with Helicobacter pylori did not develop asthma. They didn't show the reduced airflow or inflammation characteristic of asthma. And there were two other very cool things. One was that they showed that the benefit of carriage of Helicobacter was most pronounced when the mice were infected with Helicobacter strains carrying CAG-A. So CAG-A, remember, was that element, that gene that encodes for a toxin that mediates the virulence in gastric ulcers. So the presence of that virulence determinant, which of course now, knowing this, you shouldn't call it a virulence determinant.
Exactly.
It's important.
Yeah,
Yes.
It's important.
But actually, it's bad news, because I had pictured that if one could make a strain of H. pylori, which is not pathogenic but good at colonizing, it may in fact be a very good probiotic. But if CAG-A, the toxin, is needed, that falls out pretty fast.
Yeah, that's right. That's right. And actually, I have to agree, Elio. I was pretty surprised by that finding, that the benefit was most pronounced in strains that carried CAG-A. And then the other thing that they found was that in these initial studies was that the benefit was most pronounced in neonatal mice. Mm-hmm. In other words, it's important that you have Helicobacter colonization early in life.
So mice are not normally colonized with H. pylori, correct?
I believe that H. pylori is a human-specific Helicobacter, but the genus Helicobacter, at least in my discussions with Marty Blazer, the genus Helicobacter goes all the way back to the sharks. Mm-hmm. And so it is actually, it's kind of interesting to me because it's a vertebrate genus. And, the vertebrates are the only animals that have an adaptive immune system. Mm-hmm. And so it's likely, in my mind, that there's some interplay between the carriage of Helicobacter and the adaptive immune system.
They could have done a study with the murine strain of Helicobacter by taking cured animals and reinfecting them or something like that. I don't know why they didn't do it. Why go to a human, rather irrelevant organism to the mice?
You know what, Elio?
Maybe that's why they did it. It was irrelevant to the mice, so they could actually look at the specific immune responses.
I'm actually amazed that it had an effect in mice because it's not a mouse bacterium specifically. Right. And if there are mouse versions, maybe they simply aren't effective, right? Yeah.
It's a terror for folks working with rodents, and any folks working in animal husbandry in research are terrified of Helicobacter.
So do field mice carry Helicobacter?
I don't know. Good question.
So the reason I ask is because they give them this Schaechter flora, right? What they call Schaechter flora, the mice, and it's I've always thought to myself, what are those mice not getting? And it seems that they don't have their full complement.
So I've just found a paper which is entitled "Helicobacter species in wild mice found in laboratory animal facilities."
Yep.
Yeah. So I don't know if they acquired it from the lab mice but likely
vice versa.
Vice versa, yeah. So that the answer is probably yes. Yeah.
Well, have they found that there are problems with the carriage of Helicobacter in lab mice?
Yeah, I've just found a Jackson Lab page where they are really scared of it. Yes, in lab animals. So I think it's a problem. Yeah.
Do they have a reason? I guess that's my question. Do they have a reason to be scared? In other words, has it caused a problem in the past?
Let's see.
It's too bad none of us work with mice because we could all answer that question.
Yeah. Well, it's interesting because I was at a meeting recently where Dan Littman was talking about the Jackson mice versus the Taconic mice.
Yes.
And he was
Yes.
I guess the taconic mice have segmented filamentous bacteria in their gut,
That's right.
which is really important for the tuning of their immune system is different.
It's hepatic disease that it takes out their liver.
Mm-hmm.
Little is known about the epidemiology of Helicobacter hepaticus infection and the pathogenesis associated with hepatic disease, most likely transmitted fecal orally.
Mm.
So, Margaret, the fellow who did that work in Dan Littman's lab is here in my department now.
Oh, great.
And so we will get him on TWiM sometime, and he can. That would be fun, right?
Yeah, that would be
talk
super.
About. Yeah.
But the other interesting thing, this was a paper that was presented at the general meeting of ASM that got some play in the popular press about Helicobacter being implicated in Parkinson's. So here we have Helicobacter causing good, preventing asthma. Then we know about the Helico causing bad, the ulcers. And then we have this inference, and I guess you use the word, Margaret, correlation of Helicobacter causing, if you will, Parkinson's. And so the issue is whether it's poor Helicobacter doing this or whether or not it's the dynamic equilibrium in the stomach, and we just don't know who else is there besides Helicobacter, because the great claim to fame for Helicobacter is, of course, that Nobel Prize-winning experiment in which the pathology resident in Australia drank Helicobacter and proceeded to cause the pathology in his stomach that was responsible for ulcers.
Mm-hmm.
About this paper, Margaret, there is something that puzzles me. Why asthma? Why is this not a generalized reaction towards the innate immune response? I mean, is this the only thing that it can't be the only thing that Helicobacter does, namely lower the incidence of asthma? This has got the Helicobacter is in the stomach, asthma is in the lung. I mean, what's going on here? This is a broad interaction with the immune system. So I wonder if they look at anything else.
Yeah, so it's really interesting because what they did was they asked how in the world does Helicobacter have this effect? And it seems to do so by modulating the effects of the immune system. And so in essence, it calms the immune system down, what we call tolerance. And, it tames things. And they identified a specific set of cells responsible for conferring this tolerance. Most importantly, a set of immune cells called T regulatory cells. And these protective T regs, as they call them, accumulate in the lungs of neonates that have H. pylori and in the activity of other immune cells like dendritic cells, this other type. And if you deplete them from the lungs, the protection against asthma conferred by Helicobacter goes away.
Right.
So the interaction of Helicobacter and the Helicobacter's influence on the behavior of these Tregs. Now, what Elio is saying is the Helicobacter is not in the lungs. So what must be happening is that the presence of Helicobacter in the stomach causes a systemic change that influences the lungs.
But what else does it influence? That's my question. It can't just be only the lung. Why not the intestine? Why not something else?
Well, so
the respiratory system.
Oh yeah, absolutely. So it sort of goes back to, in my mind, Jeremy Nicholson's work. Mm-hmm. What Jeremy Nicholson has shown, of course, is that bacteria that are in the intestine and in various other places produce products, and those products are taken up by our circulatory system. And if you take a sample of blood, a large portion of the chemicals that are in that blood sample will be microbial in origin. And what this means to me is that every cell in your body that is serviced by blood, which is most of them, of course, is influenced now and has been over evolutionary history by the interaction with microbial products. So that's extremely powerful.
And it's all that slicing and dicing of the antigens down to the 12 mers that then get processed by the immune system. And I think the authors allude in this paper towards the end of their discussion by cautioning us by telling us that several viral and parasitic pathogens, including influenza and helminths, have been implicated in protection against asthma and allergy. So I think it goes to what Elio is saying, is that this is a complex issue with the microbes in our immune system. And I think what's elegant about this paper is they were able to tease out one microbe contributing because of the correlation with Helicobacter being depleted from our population over time, with a concomitant rise in asthma, which we know is a complex immune dynamic.
Right.
And so this may be the first paper to help us begin to tease this apart with the Tregs and the dendritic cells, because I think it's probably a combination of the Tregs and the dendritic cells that are facilitating this interaction, because I think Elio's right. Yeah. There's got to be something else going on.
Oh, yeah. It's extremely complex.
I was talking to a friend of mine who pointed out a case along these lines, which blew my mind. It turns out that if you treat people for parasitic diseases in their intestine, and if they happen to have leprosy, it exacerbates the leprosy. It goes into this violent stage of tissue destruction, which apparently the parasites keep it down. Oh, so yeah. It's this. Margaret, you last time brought us into a different mindset, and the mindset that bugs can be good for you.
Yeah.
Okay. The same bugs that in one setting, or relatives of the bug in one setting, cause cholera, vague relatives of them cause a symbiosis with squid.
Right.
This is really a powerful thing. And I think this is the take-home lesson, is that it's complicated, and even bad bugs can do good things.
Right. In this case, it supports the hypothesis that Falco and Blazer had, that losing our native co-evolved microbiota, as we are through practices in the developed nations, is responsible for an increase in asthma and likely other allergies and other big problems, as, like I said, obesity and acid reflux disease and so on. One of the things I think that's fun is that some researchers think that we should now begin to think about colonizing infants with Helicobacter at birth. And then later, should gastric ulcers or gastric cancer become an issue, treat with antibiotics.
Yeah.
But it
is a probiotic, which is what I was wondering about.
Yeah, it makes sense, although I'm not sure people would like to colonize their kids with H. pylori.
Right. Well, we have the diagnostic. We have the diagnostic because we know in the population that Helicobacter has this wonderful gene called urease, where it will actually cleave urea. And so what you do is you drink some urea and you blow in a tube and you look for the evolution of the ammonia gas. And so this is an easy clinical trial that if you want to go through it and colonize with Helicobacter and then monitor over time the evolution of the amount of ammonia that can be given off, and then correlate that back to the development of first ulcers and then secondly stomach cancer. I think we know Helicobacter is implicated in ulcers and stomach cancer, but we don't know to molecular certainty is the trigger. There may be a genetic component in the host that triggers the cancer-causing aspects, and we now have much more information than we did when we implicated poor Helicobacter in stomach cancer and ulcers.
So is my question is, are there other bacteria in the stomach besides Helicobacter?
Yeah, so Marty Blazer, Relman, and a few others. I believe there's about 100 phylotypes. Yeah, that's my recollection.
It sounds about right.
So could it be that the presence or absence of Helicobacter early in life influences the composition of that community as well, and that's important also?
Oh, yeah, like ecological succession. Yeah, that
might be worth looking at in maybe even these mice with and without Helicobacter. See what the changes in the population. That's pretty easy to do.
Yes.
Especially with now that we have, we have Ed Baelish and his notobiotic colonies, and we can, of course, do some of those experiments, especially now that we have transgenic animals, that we can be able to do some of these things with the human variants of Helicobacter and a human variant of an immune system in these animals, because you can adapt those animals for notobiotic growth.
Right.
Boy, that's a good point.
This is very interesting. I thought this was great, and I really like the way that you take the epidemiological correlations and then you do an experiment.
Oh,
You can't do it in people, but this is, pretty good.
Yeah. I think it's really important that we look for as many models as we possibly can of interactions of microbes, bacteria, viruses, and fungi, and so on, with animals to try to shed light on some very basic principles. It's a huge growing field, and I think we have to take advantage of all the experiments that evolution has done.
Yep.
Great point.
Yes. All right. On that note, let's answer a few emails here. First one is from Steve, who writes, as a microbiologist, I enjoy listening to your broadcasts. However, I was very disappointed on the one about salmonella in cantaloupes and E. coli in Lebanon, Bologna. Many incorrect statements were made, such as Salmonella enteritidis is the species. The species is Salmonella enterica. Also, it is not necessary for Salmonella to grow in food to cause illness, as was stated by one of the guests. Many foods that do not allow the growth of Salmonella, peanut butter, dry cereal, etc., have caused outbreaks of Salmonellosis. FDA does not regulate plants that make bologna. All such meat plants are regulated by USDA. Also, it was obvious that the guests knew very little about how fermented meats are manufactured and what factors are involved in destruction of E. coli 0157H7. It actually is a combination of pH, heat, and time, not just pH. The basic problem with this program was that none of your guests was a food microbiologist, so most of the conversation involved a lot of guessing. In the future, I strongly recommend that you include guests that have expertise in the area of microbiology the program is addressing, so that they know what they are talking about. Looking forward to more high-quality TWiM programs. So yes, we realize we made a number of errors there. The idea of having an expert for every topic is a great one, but practically it's difficult. So we just have to try and get it right as much as we can, because we're here on TWiM. We're developing a core of participants who will regularly be on the show, and if we have an area like this again in the future, we'll try to get an expert on, but we try and do most of it ourselves. Is that a reasonable response, Michael?
I think so.
Okay, the next one.
I appreciate your candor. You could have sloughed over this and not read it. Good for you.
No, I read all the emails we get. Yeah, and I recognize we make mistakes. I'm very interested in this field because in virology, on my other podcast, we are not experts in all viruses yet. We can talk about all viruses, but microbiology seems to be different. There seems to be a lot of specialization that prevents general talking. So I'm trying to overcome that anyway. I think we can talk about everything, but we can't say we're experts in everything. Certainly we can ask questions and say we don't know the answer to this if someone does, but I appreciate criticism for sure. All right, the next one is from Jim, who sends us a link to a low-cost sensor that can diagnose bacterial infections. This is neat in that a flatbed scanner is used repeatedly to generate time-lapse results that help in the identification of color changes, and perhaps another example of simple technology that can be applied with less training. So a quick way to identify infections by smell using a low-cost array of printed pigments as a chemical sensor. So this can identify bacteria in a few hours. Thank you, Jim. We'll post that in our show notes. The next one is from Kendall. First of all, I'd like to say I really enjoy your podcast. It's both informative and easy to listen to. I listen to it when I go for my long runs, and it makes them go by so much faster. However, on one of these long runs, I happened to see an oil sheen in the river next to the trail, and it got me thinking about oil-degrading bacteria. I don't know much about them, but I would really like to learn more, and if possible, I would love to see a TWiM about oil-degrading bacteria. You could even tie it in with the one-year anniversary of the Deepwater Horizon oil spill and talk about the impact the bacteria may or may not have made in the cleanup efforts.
Bacteria love carbon, and oil is full of carbon, and we learned from the Deepwater Horizon spill that the microbes were recruited. They did a bang-up job, and I think we're beginning to see some of the papers come out. There was that one in Science that Terry Hazen was on, in which I guess the take-home message was, yep, the bacteria came, the bacteria ate, and the concentration of oil went down. The issue was there was so much.
Yeah.
And the bacteria are so small, and of course you had to recruit, and then you had to amplify up the number of microbes in that dilute environment. And of course they were always looking for a limiting nutrient, whether it be nitrogen, whether it be phosphate, whether it be sulfur. They probably had enough sulfur from the oil, but they were probably looking for phosphate and nitrogen in order to help.
How about iron? Your favorite metal?
Old iron. Vincent always yells at me. I have this predilection for iron, so I was being good.
The battle over iron.
I was being good.
It's okay. I like it. We could certainly get someone on to talk about this, right? There are a number of people.
Oh, sure.
I actually asked Ron Atlas, and he... around the time of the anniversary of the spill, I asked Ron, and he was away, but he had recommended a few people, so maybe in the fall we can get them on. That would be interesting. All right, let's do the next two from Joe. The first one, enjoyed the last TWiM and thought of a great marketing idea for you all. 2 nesting buckets with the TWiM logo on one side, and I should have listened to TWiM on the other side. That would be for the 2-bucket disease, right, Michael?
Oh, yes. Yeah.
May all your bugs be good ones. All right. Thank you, Joe. And he writes again, he's writing to you, Michael, here.
Oh, my.
As a chemical engineer who has learned more than his share of physical chemistry over the years, I really enjoy your facility with the electrochemical aspects of biological systems.
Hey. Yeah, we noticed that too.
Yeah, I took my page from one of the original physiologists on this program, our esteemed colleague, Dr. Schaechter.
Oh boy.
It's very important.
I am hoping you can provide some insight into something that has always seemed to be a paradox to me. Note the guys on TWiV seem to be somewhat math phobic, so you are my last hope.
Oh, God. No pressure.
The third law of thermodynamics regards the property of entropy and basically states that the amount of disorder in any system must always increase. This makes sense to me as an engineer in that there is always lost work or waste energy. Heat, friction, chemical waste, etc. generated. While living things obviously obey the third law, it has always amazed me that life processes seem to be about creating order out of disorder in incredibly complex ways. The more I listen to TWiV, TWiP, and TWiM podcasts, the more striking it becomes. Viral proteins that self-assemble, DNA and RNA that form machines to make more DNA and RNA, flagella, motors, folding proteins, viruses that infect caterpillars and make a smell that attracts wasps to get a flight to a new host, ion-selective membranes to send signals in cells, evolution. This is the stuff that makes science fun. I can appreciate that living systems consume high-quality energy sources to create their ordered systems, but the whole thing seems to counter to the idea of entropy that I feel like I'm missing something. So my question is, do you have any insights into entropy and biological systems that will illuminate my understanding and answer the age-old question of what's up with that? Thanks as always for all the great science. Here you go, Michael.
Well, the short answer, it's a math thing. It's all about mass balance, and being the chemical engineer that you are, Joe, you can appreciate that microbes were the original chemical engineers, and they do mass balance very well, and their mass does indeed balance out, and the laws of entropy are indeed followed, and it's because they're so efficient at obtaining energy and processing it And they've had a few more billion years than we have to evolve their systems, that they've been able to make the best of the laws of thermodynamics and get things to go. Someday I'll talk about how they get around Henry's law, which is where they can take a gas like a volatile organic carbon like trichloroethylene or gasoline vapors and pull it out of the vapor phase, put it back into a liquid phase, and use that as a carbon source. For the microbe, it's all about energy, and they'll take anything, and so they sort of cheat. And the trick is it's really their surface to mass ratio. They have so much more surface area that they can bring in the carbon and energy into their systems.
And isn't, Michael, a matter of coupled reactions?
Absolutely, because it's the driving force.
Yeah.
Thank you. And that's how they get around things. When we first learned glycolysis as microbiologists, we were taught that the first thing that has to happen is you have to change glucose into glucose 6-phosphate. And that sort of gets around one of the first laws, because you get around diffusion because you no longer perceive glucose being concentrated against a gradient because it's no longer glucose. Mm-hmm. And so they sort of cheat the system. And it's really all about mass balance. When you total everything up, it's all there, but the microbes are really making best use of the system. I guess you could also look at them as really good accountants.
Huh? Wow.
Very good.
I'm learning by the ton.
All right. Well, I'd like to hear that explanation someday in more detail, Michael. That would be great. There are many ways you can find TWiM. We're on iTunes. We're at the Zune Marketplace. And if you do subscribe at either place, especially iTunes, please leave a comment if you haven't done so already. Just a few words you like the podcast, and that helps us to stay on the front page of the medicine podcast directory there. TWiM is free for everyone, and the way you can help us is to go over to iTunes and leave a comment. We also have an app for your Android device or iPhone. You can find that at microbeworld.org/app. As always, send us your questions and comments to twim@twiv.tv or go over to microbeworld.org/twim, and you can leave a comment there as well. Michael Schmidt, thank you for joining us once again.
It's always fascinating, Vincent. We keep coming up with cooler things to talk about.
There's unlimited in this field. There's never a shortage of good stuff. Thank you so much. Elio Schaechter from Small Things Considered, thank you for joining us today.
Oh, my pleasure. As I say, I'm learning by the ton.
Me too. I certainly am. And Margaret McFall-Ngai at the University of Wisconsin, thank you so much.
No, you're welcome. It was a lot of fun.
And I appreciate your persistence in getting a good wireless signal today.
You know what? I've just discovered something, that I should always Skype in the middle of the building instead of in my office.
Great.
So it's a good thing to find out.
I'm Vincent Racaniello, and you can find me at virology.ws. I would also like to thank the American Society for Microbiology for their support of TWiM, in particular Communications Director Barbara Hyde, and Chris Kondayan and Ray Ortega for all their behind-the-scenes work. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology. All right, everyone. Thank you so much.
This was a good one.
This was a good one. Great job on that paper, Margaret.
Yes. Thank you. Yeah. It's right up my alley, so
excellent.
It was pretty fun.
Well, you know a lot.
Thanks, everyone.
And Vincent, I put a link to Stanley and Marty Blazer's review, because it really puts it all into perspective.
That's a great idea.
Blazer,
it's a great article.
Yeah, it's a great idea. All right, everyone, have a great weekend.
Okay.
All right, thank you. Bye-bye.
Bye.